Good afternoon. I'd like to welcome you to Mindscope University, Mindscope U. We're going to give over the next half a year, Clay and I will give a series of 12 introductions. It's called an introduction, 12 easy pieces. We'll give an introduction to the brain, particularly the visual brain, and to the idea of computation and information processing in the brain. And this is really meant to serve as the intellectual background for the program that we're about to undertake, or that we've started to undertake, that we call Mindscope, that's ongoing over the next 10 years, that seeks to explore the cortex and associated systems in the mouse, and where we seek to study its structure, its function, and link the structure to the function using computation, using modeling. So all the themes that are going to be apparent over the next 10 years will be discussed by us over the next 10 years, over the next half a year, sorry. Just to give you a feeling for the lectures, could I have next slide? So we'll alternate. It's going to be roughly every second Monday, give or take a few. And so you can see we start off today with a walkthrough through the visual system, and then we'll deal with what's meant by computation, particular computation in the brain. We go to structure of cortex, computing in the retina, the optical imaging of brains that we plan to do here, functional brain imaging, information processing in the brain, what is the connectome that's going to be key to our project, seeing the world in colors, learning in cortex, visual attention and consciousness, and then finally we'll end up again with Clay, optogenetics and how to manipulate neurons. So now without further ado, let's have Clay Reid give the first lecture.
I thought I'd start this talk with what attracted me to study both the brain and the visual cortex. More than 25 years ago, when I started graduate school, almost the first thing that I encountered was this. First, you can almost just close your eyes and listen to the brain. So this is the sound of one neuron inside a brain that consists of perhaps 1 billion neurons. We have many tens of billions of neurons in our brain. And it sounds almost like a Geiger counter, but it also sounds a bit like a very fast Morse code. And this is how the brain communicates with itself. This is a single neuron that's firing an action potential, a spike, we sometimes call it. And it's the unit of neural activity. These individual spikes, and we have perhaps 1 billion of those happening in a second, are going on inside our brain, and it's in a language that we really don't understand. And especially if you're just listening to a neuron, you certainly don't understand what it's saying to the rest of the brain. For the first thousands of years when people were thinking about the brain, this sound was completely inaudible. By the 1920s, though, when amplifiers began to be made, and certainly by the 1950s, when amplifiers were getting quite good, this sound was heard for the first time. And so in the 1950s, it was heard for the first time in the mammalian brain. But this is what I saw at the same time that I heard a neuron, precisely this kind of neuron, in the laboratory of Robert Shapley at Rockefeller University. This is a movie made by Hubel and Wiesel, two scientists that I'll tell you about later. But now let's see if you can understand what that neuron is doing just a little bit. We're listening to the neuron, but we're looking at what the animal was seeing when that neuron in the middle of its brain was firing, and see if you can figure out roughly what that neuron is doing. Right now it's pretty clear. That neuron, actually it's firing a bit less right now. And now it's firing when the light turns off. So at first we thought the neuron was a simple light detector. It turned on whenever that light was in the right place. But in fact, it's a slightly complex slightly complex thing that at most simple, it is a light detector. It's like a pixel in a TV camera. But clearly the retina, so this is, you're listening to a neuron in the middle of the brain that's getting its dominant input from the retina. But clearly that neuron isn't just seeing a point of light. It's seeing a point of light, but it's actually inhibited by light outside of that central region. And we have roughly 1 million of these neurons in each one of our eyes. And the very smallest of those see the very finest details that we can see, for instance, a star. So these neurons are great at seeing a star, but that very same neuron, if it moved along and looked at the moon, it would be less excited because it's very good at seeing small things but not big things. And we like to say that it senses contrast. It doesn't sense mean light levels. So if you turn the lights on in a room, it fires a little bit, but if you show a small light in a very small, in a large dark room, then that neuron will be optimally excited. So you can see why the visual system is a wonderful place to do neuroscience. It really, from the very beginning, you can understand at an intuitive level, because we are such visual animals, you can understand how visual neurons work. And really, the visual system is a wonderful place to study the brain because its connections to the brain are so radically obvious. With any amount of introspection, you know that you're seeing through your eyes, and then the question comes up, what happens to the information that goes from your eyes into the thing that thinks? And people had various theories of how the brain was put together, what part of our body we thought with. It might have been the heart, but most people, even in times of the Greek philosophers, thought that the brain is likely to be what we think with. It wasn't until, though, roughly the year 1000 that someone figured out really how the visual system works. And this is a great philosopher and scientist, Ibn al-Haytham, or in older literature in English, he was known as Alhazen, born in 965 in what's now Iraq and died in 1040 in Cairo. And he was in the tradition of the great Arab philosophers, that's in the 700, 800s and beyond, who revived the Greek philosophy and kept alive the traditions of antiquity. But a few of those philosophers were also scientists, and I think Alhazen is probably the greatest of those. And he's thought of as the founder of modern optics, of modern experimental physics because of what he did with optics, but for our purposes he was the founder of physiological optics. There were a bunch of theories in ancient times about how the eye saw, and many of the theories involved things coming out of the eyes, touching the outside world in some way. But Alhazen, really from his experiments in optics, he was the first person to understand a camera obscura, which is a dark room with a pinhole in it and that forms an image. He was the person who really did the first experiment to understand that. And from those he understood that it's the eyes, here's a picture, the eyes that are simply a camera. And he understood that an image of the outside world is formed on the eye. But he also was a keen observer of anatomy and saw that these cables from the eye go into the brain, cross over, which is a real insight, and then go to something which is inside the brain. This isn't the greatest diagram of the brain, but it's something I actually even started my thesis talk with. It's really an early example. And recently I've read in a number of places that people consider this the very first diagram of the brain, or probably more accurately, the oldest surviving diagram of the brain. And I think it's not coincidental at all that it is, the ideas came from vision. This is a modern view of the brain and the visual system from David Hubel's book. Again, I've spoke of David Hubel and Torsten Wiesel, two scientists who started at Johns Hopkins and then soon moved to Harvard Medical School in the early 60s. And from their time at Harvard Medical School mostly, they really unlocked many of the secrets of the visual cortex. And I'll be talking about their work a great deal. But here's just a simple diagram of the human brain. And the dominant feature, I guess because David Hubel studies vision, is not just the eye, but actually exactly the setup, the experimental setup that you just saw from Hubel and Wiesel's experiment. A spot of light on a dark background that goes from the outside world, is projected on the back of the eye, and then the interesting thing is, what is the transformation that goes from light to nervous signals, the signals that you heard at the beginning of the talk, and how are these signals propagated through the brain? I'll just tell you a few facts of neuroanatomy. The first neurons that light encounters are in the retina. The retina is actually part of the central nervous system of the brain, where signals are first, again, turned from light into nervous activity. And there's a fair amount of processing that goes on in the retina, as you saw already. This center, surround, contrast-sensitive responses are created in the retina. And then that information from the retina is sent to a structure in the middle of the brain, which is the thalamus. And the thalamus and cortex, then here's the cerebral cortex on the outside of the brain. The entire brain that you see, or most of the brain that you see on the top, and the part of the brain that's most expanded in humans, and I think really it's what makes us human, is this great expansion of the cerebral cortex. And I think you'll hear a lot in this course, but certainly at the Institute, we are beginning an endeavor that really is trying to understand the thalamocortical system and how it processes information in the visual system. So here we have three things again: retina, thalamus, and visual cortex. And I'll be exploring what goes on in all three a bit. The thalamus, I might say LGN. The LGN is it's the visual thalamus, the part of the thalamus that transmits information from eye to brain. And it's important to note also that the thalamus is not the only target of these neurons that are in the retina. There are other targets, such as the colliculus right there, and other targets within the brain. So you'll see not so many pictures of neurons in this talk, but I think it's important to show this one picture of the retina. This, again, has the neurons that translate, really, from photons, from optical things, into the neural language. And a neuron is a brain cell. There are several types of cells in the brain, but we'll be discussing almost entirely neurons. Right here, in this layer of the retina, at the very top, are known as photoreceptors. And there are two types of photoreceptors that I'm sure everyone has heard of: rods and cones. The rods are what we use to see in dark scenes. They're the ones that allow us to see stars. The cones are what we use when the light is bright, whether it's in a room like this that's lit pretty well, but certainly in daylight. The cones are what allow us to see. So most of vision is done by the cones. Photons excite this part of the cell. There's a machinery in this part of the cell. And right here is what's called a synapse. A synapse is the thing that communicates from one cell to the next. So this is the presynaptic cell, and that's the postsynaptic cell. And while most of signaling within a cell is electrical, most, but not all, of signaling between neurons is chemical. So these photoreceptors go to the next level, which are called bipolar cells. And I won't say much about them. They really are just the cells that transmit, for our purposes today, transmit information. And now we're in neuron land, we're in the brain, information from photoreceptors down on to the cells that are the output of the retina, the ganglion cells. So we have really three cells you need to know about: the photoreceptors, the bipolar cells, which to first approximation are a relay, but to second approximation are very rich computing devices that get multiple inputs from different types of cells I won't discuss. And then finally, the ganglion cells that send their axons. The output of a neuron is an axon. And whenever a neuron spikes, makes those action potentials that you heard at the very beginning, that information can travel long distances within the brain along a nerve to go to the next part of the brain. So light in, action potentials out, and those action potentials travel from eye to brain. A funny fact about the eye is that it's built backwards, or the retina is that it's put in the eye backwards. It's slightly perhaps advanced for this introductory course, introductory lecture, but I'm using that fact to make a point. Light must go through the other cell types from the top in order to hit the photoreceptors. So it looks like this. The light that we saw flashing on the retina was in fact traveling all the way through the retina, through all the cells in the center of the eye, the middle retina, to be focused onto the photoreceptors. And then neural signals are sent the other way. That's interesting in and of itself, but it's very interesting and lucky for neuroscientists nowadays just to note the fact that brain tissue, especially the retina, but all of brain tissue, it looks sort of cloudy when you look at a lot of it, but when you look at very thin slivers, so the thickness that the light has to travel through the retina is roughly the thickness of 2 human hairs. It seems like nothing, but it's a lot of brain in between, within the span of 2 human hairs. But the eye is essentially transparent in that distance. And as you see, as we're getting into optical techniques in current neurobiology, it's important that the brain is not opaque, but it's fairly transparent, especially for red light. But that's another story. So I thought I've showed you a bit of neuroscience. Let me show you a few more movies because they're fun and because they really indicate the amount of computation that goes on within the brain. So you already get the idea that a fair amount of transformation goes on in the retina. Those transformations are transmitted to the thalamus. But then in the cortex, that's when things begin to get far more complex and, at least for me, far more interesting. I'll be showing you 2 characteristics of visual cortical neurons that are not terrifically complex, but have kept scientists quite busy for the 50 years since they were discovered by Hubel and Wiesel. The first one is orientation. And let me just play the movie. So we're listening to the action potentials from one cell in the cortex, and it's exactly the same game. Use a light stimulus to try to find out where the neuron is looking. So the animal is looking at it, but this one neuron in the animal's brain is looking at a very specific place out in its visual world. And this is a little bit complicated, so it's useful for them to draw. So this is, again, Hubel and Wiesel, probably in the late 60s, making a video. It's useful to draw a box where light will excite the neuron. And as far as we know so far, it's just a light detector, like the retina. See? You put that bar through it, and it gets excited by light. But they're being a little bit cagey. They're not showing you what's very special about this neuron. But here comes the critical experiment, and I'll be quiet, except notice the orientation changes. And now watch. It fires. Here's the really critical experiment. Watch this. It was firing like a mad Geiger counter before, and here it is back again when Hubel and Wiesel were showing the appropriate orientation. So this cell isn't just looking at light within its little box that's called its receptive field. It's looking at patterns in the visual stimulation. And that poses the question, how does it do it? And the other question, why does it do it? Let me show you another one because I'll be getting back to it later. Direction. They made it noisy back then. So here's just another point of light. And again, they're being cagey. They're just jiggling it back and forth and drawing a box which is the spatial receptive field. It's where the neuron is looking. But pretty soon they'll perform the critical experiment, and you can probably guess by the title what it is. They drew the arrow, cheating. Pretty amazing. So this neuron not only cares about orientation, which they didn't show, but it also cares about the orientation.
The direct direction.
Thank you. That was a question. What did you mean by orientation? I meant direction by orientation. It cares about the direction. And let me go back to the previous slide because I neglected to say something. You know, what we're interested in are two questions. And depending on who you are, you might pose the questions in different ways. The first one is, what does a neuron do? And you've already seen what three different neurons do. One is a detector of just light with a little bit of contrast thrown in. One is a detector of orientation. And the third one is a detector of the direction of motion. So I'd put it that way. Other people, and I would also perhaps put it in the way, what is the computation? How does the brain computer, to use a common metaphor, do this? And what's kind of fun is, you know, I'm an experimental physiologist who cares a great deal about neural computation, and Christoph is a computational or a theoretical neuroscientist who obviously cares a great deal about physiology. And these are the two questions that we and our colleagues ask, and they're really interchangeable. A second distinct question is, how does it do it? You know, how does the machine do all these complicated things? And another way of posing that same question is, what is the computation? If we take all of the neurons that we know about, that we're listening to, how does the circuit in which they're embedded perform these complicated computations? So we jumped from 1,000 years ago to 50 years ago. Let's jump back to roughly 100 years ago to, I think everyone would agree, is the founder of modern neuroscience. And that's Santiago Ramon y Cajal, who was an anatomist. And he was lucky enough to live in a time where other scientists created methods to make what used to be invisible, visible. So Hubel and Wiesel were lucky enough in their time to be living at a time when what used to be silent became audible. Ramon y Cajal lived at a time when it wasn't just microscopy, it was the staining of neurons, allowed things to become visible. And the discovery that he based his work on was by an Italian neuroscientist, Golgi, who figured out a way to make neurons, individual neurons, black. And most of their neighbors were invisible. And neurons are such complicated things that if you're looking at all of them, you see nothing. But if you look at one on a clear background, you see its structure. And I said that Cajal was lucky enough to live at that time. But of course, he was, you know, one of the great scientists, that, you know, a true genius that saw what people, that, you know, tens of people, maybe hundreds, were seeing at the same time that when he worked. But he understood it in a way that was revolutionary. And again, you could say the same thing of Hubel and Wiesel. There were many people listening to neurons, but they understood what might be important in cortical computation, or they wouldn't call it that, in cortical physiology back then. So what did Cajal see? He saw the neurons very clearly. You know, for instance, right here, I've already shown you the retina with photoreceptors, bipolar cells, and the ganglion cells. Other people had drawn similar pictures then. What Cajal did, so here we have the retina, and we have the nerve exiting the eye, the optic nerve. And within a nerve are many nerve fibers, or axons. So this is like a superhighway. In us, we have 1 million axons, or individual wires, leaving the eye. And it seems obvious to us, and maybe it was obvious to most working scientists then, but this is what he did. Right there next to my pointer, he put arrows in the brain. These arrows were not in the Golgi stains. These arrows were what he intuited. Information has to flow from eye to brain. I think anyone would know that. But what he further did was he put arrows all over the brain. He put arrows from retina to thalamus, obvious perhaps, from thalamus to cortex, from cortex to other regions of cortex, and Christoph loves this, from cortex down to other structures, including feedback to the thalamus. I don't think Cahal's shown that, but he might have, I'm not sure. But he saw black and white, and with no knowledge of modern physiology, with no knowledge of what individual nerve cells did, he intuited that there's an arrow from here to there, one neuron to one neuron, and that's actually correct, and from this neuron onto multiple neurons, and that's also correct. So that really was the insight that all of modern neuroscience is based on. So here we have the two great men 1,000 years ago and 100 years ago. That, you know, in here, this is not a real picture, but he was made to look like, you know, great modern scientist by some 19th century person who was making a picture of Alhazen. But, you know, it probably was based on a real drawing, perhaps. The founder of modern optics, experimental physics, physiological optics, and the founder of modern neuroscience. So let's get back to Hubel and Wiesel, and in fact, their mentor, Stephen Kuffler, who was at Johns Hopkins and left with them in 1960, roughly, to found the Department of Neurobiology at Harvard Medical School, where I packed up from 2 weeks ago today, I guess. But anyway, from that department, they made a number of discoveries about the visual system. This was a discovery, you've already seen it, from the 1950s, where he where he discovered in the mammalian retina what had been seen also by Hartline in the frog retina about the center surround receptive field. And you've you've heard the data that I'm showing you right here. But the real reason I'm showing you this slide is to define what a receptive field is. I've used the word about 20 times so far. As defined by Hartline in 1938 for the frog retina, it's the region of the retina which must be illuminated in order to obtain a response from any given fiber, a fiber being a nerve cell or an axon. But any visual neuron has a receptive field, and this is sometimes is a point of confusion. The cells you've seen all have a region of space that excite them, but one is a contrast detector, one is an orientation detector, and one is a direction detector. I'm using the word detector very loosely because many people say that's leading the witness almost. You know, we don't really know exactly why these neurons do what they do, but it's a good shorthand for our understanding of what they might be useful for. So let me go through a bit more. So the other reason I wanted to show you this is to get you familiar with looking at spikes rather than just hearing spikes. So right there, we have time along the x-axis, and along the y-axis is just the voltage that was measured by one of these amplifiers that really revolutionized neuroscience. So each one of those dots, each one of those lines is the firing of an action potential. And when you put light in the center of the receptive field, here that light is turned on for maybe a second, you get a bunch of action potentials. When you excite all of the center, you get many more action potentials. That's what they did in the movie. They excited the whole center and it went off like a Geiger counter. But if you put a bunch of light in the surround and not the center, it's when you turn off light that the neuron is excited. Sort of paradoxical, but very important for understanding contrast. Christoph later on will tell you about color, so let me just give you an outline of a neuron in this same part of the brain, in the visual thalamus, that again Hubel and Wiesel studied. This neuron is red on, green off, but they used blue in this case, receptive field. You see that when the red light turns on, the neuron goes crazy, fires many action potentials. When the blue light turns on, it's inhibited. There's no activity. And they're so beautifully balanced that when a white light is turned on, you get very little activity as well. So it's a perfect red detector for these large red spots. So what I've shown you is examples from the retina and the thalamus and the primary visual cortex. But what makes the cerebral cortex so interesting, and people knew this from the 1880s, is that it's made up of up to many tens of distinct areas. The biggest area in our brain and in most mammalian brains is what's known as primary visual cortex. And this is the TV camera at the back of our head, where the very simplest transformations are done, such as combining the input from two eyes, such as computing orientation and things like that. Very importantly, I forgot to say it, that direction is also computed in the retina. But so there's direction selectivity in the retina, but most cortical neuroscientists think that it's recomputed in a new fashion in many brains. Certainly in a primate brain, direction is recomputed within the cortex. But even though it's the biggest area, it's not the only visual area. And a great deal of fabulous neuroscience came from really the early 70s until now, but with its peak in the 90s, of looking at all sorts of different cortical areas. The peak of the discovery. People are still studying these areas, but over that period, people discovered well more than 10, probably 20 distinct areas, all of which see, but they see distinct things in the visual scenes. And so here we have, this is a diagram that scientist Tom Albright put together. Retinal ganglion cells. There are multiple streams of retinal ganglion cells, some for color, some not for color. The LGN, or the thalamus, that have these streams. And then we have V1, where there are a large number of neurons that do a number of distinct things, for instance, color or orientation. The information for V1 gets distributed. I'm only showing probably, whatever that is, 9 areas here. But again, there are well more than 20 distinct areas that process different things about the visual scene. And in particular, there are 2 streams. One is called the parietal stream, because it's in this part of the brain, the parietal cortex. One is called the temporal brain stream of vision, because it's here by our temples in the temporal cortex. And these distinct streams are starting with temporal cortex. That's where the what neurons are, neurons that see complex details or complex features about visual scenes, and the where stream, which is concerned much more with motion, where things are in the visual world, and even with three-dimensional things about vision. I'm going to go through several examples, first of something that you've seen already, direction selectivity in the brain, but I'll show it to you in a way that's different. Rather than listening to the brain, it's possible to watch the brain in action. And this is one of the revolutions of the past 30 years, that a number of scientists have figured out ways of doing physiology with photons, rather than doing physiology by amplifying electrons or amplifying voltages, by simply looking at the brain and putting fancy fluorescent indicators on the brain that get brighter when a neuron is active. And many scientists are doing that, and my lab at Harvard was doing a fair amount of that, and will continue here as well. I'm showing you a direction selective experiment, but now so again, let's go back to the stimulus. The stimulus is very simple. It's very similar to what Hubel and Wiesel did, having bars going in different directions. But rather than listening to a neuron, let's watch neurons instead. So here we have, on the right-hand side, a picture that doesn't look like much yet. But now it looks like something. On the upper right-hand corner is the stimulus, but the rest of the picture is actually roughly 100 neurons, and you can just watch them flash away. Rather than listening to them, you can see them. So, quite literally, it's seeing the visual system see. And as you can probably see, half the neurons fire now. The other half of the neurons fire now. Some of the neurons like stimuli going down and to the left. The other neurons like stimuli going up and to the right. So rather than listening to one at a time, or maybe 10 at a time in modern experiments, we can look at 100 cells at a time, or maybe 1,000. And this is where it's fun that the transformation from photons into the language of neurons, and now back again into photons, so we can see the brain thinking. And we're using the fact that the brain is decently transparent, so we can see these things in the living brain. This is the example of the most complex receptive field, or most specific and complex receptive field, I think, in the literature. And this is work by Christoph Koch and his longtime collaborator Reid at Caltech and at UCLA, recording in a human. This is a human patient, which for medical reasons, neurosurgical reasons, the brain was exposed, and in those patients, it's common to record the electrical activity of neurons in their brain for diagnostic purposes, but also one can add on a brief but important neurophysiological experiment. And this is a human being who was looking at pictures on a computer monitor, and Christoph and Reid and collaborators were listening to a neuron. And in their eureka moment, when they were showing, people already knew that there were neurons in the temporal cortex, the what pathway, that saw faces that had been described in species other than humans. But they found, as they were showing a battery of faces, a neuron that just fired for Jennifer Aniston. They noticed it once, and then they said, oh my goodness, let's go back. Let's get a bunch of pictures of Jennifer Aniston and see if the neuron is a neuron that signals this picture of her. But no, it wasn't. It was a neuron that showed that would fire action potentials whenever this human saw a picture of Jennifer Aniston. And even when in later experiments, when the human thought of Jennifer Aniston, this neuron would fire. And that gets into both complex receptive fields, but even in the end, into behavioral neuroscience. For a human, you can say, think of Jennifer Aniston, and you can study its neuron, and study the human's neuron that sees Jennifer Aniston and find an amazing behavioral thing. That thinking of Jennifer Aniston makes this neuron with a, quote, visual receptive field fire. There's a great field, and we'll talk about it later, of behavioral neuroscience, where you try to train animals to do things. It's never as complex as think of Jennifer Aniston, but trying to do the full circle from perception to neurons in their activity to behavior is one of the great themes in neuroscience, and one of the themes that we'll be addressing at the Allen Institute for the next decade, but only for the next 6 months in this course. But now we're almost at the end. What we had was, starting from anatomy 1,000 years ago, from understanding a great deal about how the brain works by seeing the eye, and just 1,000 years ago, guessing or intuiting that the connection from eye to brain, from eye to head, had to do with what we were thinking with. We were thinking with the brain. And then 100 years ago, the detailed connections from eye to brain that Cajal, as a great anatomist, saw and could draw the arrows and really trace the pathways that now, as physiologists, we know he got it all right. Then to 50 years ago, studying receptive fields with electrodes, with the new technology of being able to listen to cells, through what I just showed you, looking at neurons, inverting the transformation from photons in to photons out. But it gets even better. Roughly 5 or probably almost 10 years ago, a number of groups, but in particular Carl Deisseroth's group, were working on proteins that sound very much like the proteins in our eyes. We have rhodopsin or conopsins in our eyes. There's a protein that was discovered in bacteria by another group called channel rhodopsins. When light hit these proteins, it opened up a neuron so that its electrical activity would change. I won't go into the details, but it's a way of transforming from light to electricity in neurons. And that's precisely what a photoreceptor does. But now with molecular biology and modern optical techniques, you can put that protein that is similar but simpler than the one that's in our eyes, and you can put that into neurons with essentially molecular biology and viruses, but one can do this with transgenic animals, which we'll discuss later on. One can put this protein into these neurons, but very importantly, this is their first Nature Neuroscience paper of Ed Boyden in Carl Dijsselroth's lab, where if they flashed light on this neuron, it fired action potentials. And now we're back in 1953. Here's the light stimulus of a neuron that's given to the neuron, and the neuron fires action potentials. So we're able, what's great about the visual system as a sensory neuroscientist, as a physiologist, is you have great control of what goes into the brain, from a point of light to Jennifer Aniston. And that is a stimulus that's very rich, and we're able to control it very well because optics are great, and frankly, the optics of our eyes are great, and we're such visual animals. Now we're able to short circuit it, and with this new technique known as optogenetics, excite any neuron in the brain with light. So in the final slide, I just want to say that it really, for all of us in the field of neuroscience, when we were stuck with just electrophysiology, there's a lot of great science going on with electrophysiology, but with the optical revolution, where we can see activity and now we can create activity with light, the classes of experiments that we can do are just so much greater. So in the same brain, obviously it's not a human brain, but it's nice to use this diagram from David Hubel. Whoops. We have vision, and I told you a great deal about visual receptive fields and how one might study them. We have electrophysiology, which I'm not showing, but the first experiment that I showed you was putting one electrode here. We're planning, we and others are planning and actually have put hundreds of electrodes in here. So it's possible to do the old-fashioned experiments in a new-fashioned way where you can record from hundreds and potentially thousands of neurons electrically. But the really new technology is the imaging, where in the same brain where you're exciting with visual stimuli, you can watch neurons flash away and study many more things, many neurons at once, many more specific things about the neurons at once. And it's also an anatomical technique, so you can see which neuron does what, and that's very powerful when you want to understand the mechanism. And then the final thing, and this can all be in the same experiment, we have vision in, we have optical monitoring out, and we have what's almost like vision in at the back of the brain. We can be flashing at individual neurons in the brain, and if we have an idea about how two neurons communicate to each other in order to make one of them see this direction and not that direction, or perhaps even see Jennifer Aniston as opposed to Ashton Kutcher or someone, we can excite one of the neurons that is important in our hypothesis and flash it. So that's where I'll stop. That really, technology has driven much of what we know about the visual system, and really the technologies of molecular biology, computers to allow us to do complicated experiments, and optics has really transformed neuroscience in a way that we and others will be using for the coming years. So thanks. It's a great question. It's a great question, and it's something that I just touched on and didn't really explain what I meant, and certainly didn't give theories. So the question is this. We have retinal ganglion cells that I told you about in passing that compute the direction of motion. And then we also have neurons in the cortex that compute the direction of motion. And I said that most of us who study primary visual cortex are pretty sure that in most species, or in many species, it's recomputed in the cortex. And the great question is not what did you mean, but why? So the question is, why might it be recomputed? And I think there's a pretty simple answer, actually. You might want to compute direction for many different reasons. In the cortex, you saw the receptive field. You saw the mapping of the receptive field for the directionally selective neuron. And one thing you might have noticed is that the receptive field was quite small. It was the same size as, it's a little bit bigger, I'll just tell you, you didn't really see it. It's just a little bit bigger than the receptive field of a retinal ganglion cell, the very smallest receptive fields. It's roughly the same size of the receptive field of an orientation selective neuron. So if one wants to say that it's a detector or it's encoding a feature, it's encoding the feature of motion of a very small thing. The directionally selective neurons in the retina are much bigger. So the spatial receptive field, which is the area in space that it can see at all, is much bigger. It might be where the receptive fields in our retina can be as small as the smallest thing we can see, a 60th of a degree. And the smallest directionally selective cortical receptive field might be twice that big or 3 times that big. Whereas in the retina, the receptive fields might be 20 degrees across. And that signal, people think, is very important for detecting large objects, and it might also be very important for orienting our head with respect to things that are moving, or more importantly, perhaps orienting our eyes with respect to things that are moving. So you can use motion to detect small things that are moving and say exactly where they are, or you can detect motion over a large area and use that signal for much coarser but probably equally important things, such as turning your gaze, turning your eye, orienting yourself, or just detecting an eagle about to grab you if you're a mouse. So big and small is the simple answer. That's a great question. These are 2 great questions about the retina. So the question is, there are a lot of cells between the photoreceptors and the ganglion cells. And I've told you or hinted that direction is one of the things that these cells might compute. Is there anything else? And the answer is many things. So there are 2 classes of cells that are called horizontal cells that communicate with the photoreceptors and the relay neurons, the bipolars. There are 20s, or there are 10s of amacrine cells that are communicating directly with the ganglion cells. We understand very little of what they do. There's just a beautiful study from the laboratory of Winfried Denk, which you'll hear about connectomics and functional connectomics. That was the first example of functional connectomics in the retina, where they had just a lovely story of how one of the amacrine cells computes direction selectivity. But that's one of 28 classes. So there's plenty to discover in the retina. Yeah, so to study these high-order things. So the question is this. If I can encapsulate it, essentially what ensures us, our knowledge as scientists, that this neuron, the Jennifer Aniston cell, is in fact a particular Jennifer Aniston cell, as opposed to encoding something as desire or something that's much more complex than Jennifer Aniston. The first half of the question was the alternative view of how this might work is that there is a network of cells, and might the activity of this network of cells be a more common or more logical way of signaling complex things. So it feeds into a long debate, which is why it's such a great study. It feeds into a long debate about distributed coding, where we might have 10,000 cells in our face area and some complicated constellation. I like using the word constellation nowadays to talk about cells because you can just think of the flashing lights. So some complicated constellation of cells might encode Jennifer Aniston, as opposed to one cell, which is Jennifer Aniston. And the debate really used to be couched in terms of not Jennifer Aniston, but your grandmother. That it was called, and really it was used as reductio ad absurdum of the idea that everything we think of is encoded by a neuron. People said, I just can't imagine that you have enough brain cells in your head to have one of them encode your grandmother. So for all the faces we know, it's very unlikely that there's one cell for all the faces we know. But perhaps if Jennifer Aniston plays a large enough role in a person's thinking, the brain can devote a certain number of cells that learn to see Jennifer Aniston. So it's a very rich debate and it falls into the category of distributed coding and specific labeled lines. And it's great to have this wonderful example where something that many people said, never, not on my watch, I can't imagine a neuron like this existing in the brain, and in fact it does. But of course, there has to be also distributed coding as well. So he's asking about the two-photon imaging. I didn't even use that word, but the optical approach of looking at 100 neurons at once. And he asked the question, two questions. One, are these neurons receiving the same input, roughly? And the other question is, can you tell anything about the system from when the neurons are activated? To address the first question, to first approximation, these neurons are likely to receive similar input. Let me take that back. This is secondary visual cortex. This is area 18. Those neurons are likely to be receiving input from primary visual cortex that encode different directions. But we don't know yet. I'm just making a guess. If we were doing the same experiment in primary visual cortex, I could say with some confidence that they were receiving similar inputs. So it definitely can be the case where neurons that do different things receive the same inputs, but they're combined in a different way. And the second question, is there anything about the timing that might tell you about multiple types of inputs? That experiment is 7 years old, and we were recording at 1 picture per second, or 1 picture every 2 seconds. So we couldn't even begin to look at the timing. And that's with modern optical techniques, we can take a picture every 30th of a second, and it really could be arbitrarily fast. And you can look at timing with optical techniques, but it's really, it brings up a good question. If you had these optical techniques, why would you ever go back to electrodes where you have these spikes? And the answer is time. That when you're using electrical techniques to listen in to the brain, you know when the neuron fired an action potential to within a few tenths of a millisecond, to roughly a 10,000th of a second. And you can use that information to really learn a lot about how the network is processing information. So we have great new techniques, but some of the tried and true techniques you have to keep because they do what they do extraordinarily well. So yet another great question, and really sort of goes to the heart of the matter. The first mini question was he assumed that in the optical experiment we're looking near the surface of the brain. We're looking at the cortex, which is on the surface of the brain, and the answer is yes. The deeper question, the second question, was how deep within the cortex can you go? I haven't told you yet, but I guess Cahal did. That the cortex, like the retina, is a structure the cortex is a structure that has several depths. There are neurons up here towards the surface, there are neurons down here towards the bottom. The whole thing in the mouse is a millimeter thick. And within that millimeter, there are classically described 6 layers from the top down to the bottom. And the question is, which layers can we see? And similar to the question about timing, with the early studies, we could only look at slow things, or at least we could. Other people could look at faster things. But now, as techniques get better, we can look deeper and deeper into the cortex. The early experiments were only done near the surface of the brain in what's known as layer 2-3. But now, with selective staining, better lasers for the microscope, and other factors, we're able to look through layer 1, 2, 3, layer 4, and even layer 5, and do this kind of physiology. The very bottom layer, layer 6, as you're approaching a millimeter, it's pretty hard, but I think we and others will get there. So what used to be a surface technique for just the top of the cortex is going down to most of the thickness of the cortex in small species like mice. You can't see the thalamus, which is another reason, unless you do complex and sort of difficult things. But for the cortex, you can see most of the layers of the cortex. That's probably a good place to stop. So, thanks.